Conor, today on Discovery Matters, our guest is working at the cutting edge of biology and artificial intelligence. Some of the stuff that he's doing sounds like science fiction.
Everything sounds like science fiction today. It's just, you cannot believe what's coming around the corner next. It just gets stranger and more fun.
It does.
What are we looking at? Strange or more fun?
Well, we're actually thinking very, very small, microscopic innovations that could change the way we understand life itself.
This is hugely beneficial to researchers, and researchers will benefit the most from the revolution that we're seeing.
Okay, now I'm intrigued, and that is what matters on today's episode of Discovery Matters.
Conor, meet Samuel.
My name is Samuel King. I'm a third-year PhD candidate at Stanford. I research bioengineering, broadly AI methods for biology.
Samuel works on both the experimental and the computational side of artificial intelligence.
So explain that to me in words of one syllable.
So not only just doing things like on the computer, but actually translating them into real life and testing them in the lab.
Okay, so how did he get to this incredible intersection of AI and biology?
When I was younger, I definitely had no idea that I would end up in biology. I actually pursued a lot more artistic endeavors growing up. I played a lot of music. I did a lot of drawing and painting. I grew up in a very, very small rural town in Eastern Canada, and my parents work in the fishing industry, and my high school didn't really offer very many science classes at all. But I did take a biology class at my high school.
It was there in the classroom where Samuel grew deeply curious about biology.
And I would constantly ask my teacher many questions, and she would just tell me, you have to go to university to find out the answers to those questions. And I was thinking, okay, maybe it makes sense for me to go study biology because I just seem to be like weirdly curious about it, and I'm not sure why.
Another motivator at the time was that it was very easy to see the effects of climate change in Samuel's rural community.
It was really impacting the fishing industry that my parents work in. And I, for whatever reason at the time, thought that studying biology might help me understand ways that we can pursue solving problems in climate change. And I really love the idea of biotechnology. I think another part of me that was really drawn towards AI was the way it's completely transforming how we see biology. And I see it as like a better path towards ultimately solving those problems by developing these methods that really accelerate biology and ways we can create new biotechnologies. And, I just love being in a place where I can work on many interdisciplinary things, and being at the intersection of computation and experimentation is a really good place to be as a biologist right now.
The reason why we're hearing from Samuel today is because last year, using AI, he and his colleagues produced functional viral genome designs and used them to create bacteriophages that can kill drug-resistant bacteria.
We used a very safe laboratory strain of E. coli called E. coli C. This is the host strain for a very classic bacteriophage called Phi-X174, which is the first DNA genome ever sequenced. For decades, people have been studying Phi-X174 to understand a lot of basic things about how genomes work. We used the strain E. coli C as our host strain for the generated phages because we were trying to generate phages that looked like Phi-X174. And since we don't really have the evaluation metrics to determine what that actually should be, we had to work off of what we do know, which is what a natural bacteriophage looks like. And so Phi-X174, because of its historical precedent and safety, made a lot of sense as kind of a reference point for what we should use to determine our design criteria. And biological language models have, through seeing so many genomes and so much of evolution, have kind of learned a fitness landscape almost around beneficial or non-beneficial mutations. Like, for example, we can use the model to actually score how detrimental a mutation might be to a gene. And we validated this on actual experimentally determined data sets, and the model has a pretty good understanding of when a mutation is actually harmful to an organism or not. And so it seemed like in the generated phages that worked, the model must have designed new mutations in them that somehow benefited the fitness relative to phi x174. And that's what we saw in our experiments.
And I just love a phage. So he's using AI to design viruses.
Yeah.
Amazing.
So the word virus is pretty scary, I think, in our society. It's broadly viewed as a very negative thing. And of course, like viruses cause great harm to the world through pandemics and many infectious diseases and sicknesses. But viruses also can be used in many good ways. They're very effective at what they do. They're these small little genetic entities that package a small genome that has just really the minimal components necessary for it to get into a host cell and replicate. And being so evolutionarily streamlined, they've become really, really efficient. And in biotechnology, we've been able to learn from this and then leverage viruses for many useful purposes.
A great example of this is viruses that infect bacteria.
From the ancient Greek phagein, to devour, and bacteria, of course.
We use them every day in molecular biology for a whole range of things, just basic studies in understanding biological molecules, but also in applications like therapeutics. So like bacteriophages can actually be used as therapeutics or treatments to fight off bacterial diseases, especially like antibiotic resistance. And they're also used as little packages to engineer bacterial cells and use those for different purposes, like in research or in therapies or biotechnology.
So we've talked before about how viruses are used to carry good medicines into the body, viral vectors, for example. But I've not yet heard about AI being used to design an entire bacteriophage itself. So can you give us a little breakdown on how this works?
There have been a lot of advances in large language models. So, things like ChatGPT, but instead of trained on text from the internet, we've been able to train these models in biology on text like DNA sequences or protein sequences.
He's a member of Professor Brian Heath's Laboratory of Evolutionary Design, and they created large language models trained on millions and millions of genomes from all domains of life.
Not just things like bacteria or archaea, but human genomes, plant genomes, animal genomes, and bacteriophage genomes. And so we wanted to see if these language models understand DNA well enough that they could actually design an entire genome or generate an entire genome at once. And it made a lot of sense to do that with bacteriophages because all of these useful purposes that I mentioned earlier, but also because they're very feasible to test in the lab, and there's decades of experimental precedent in their safety and protocols on ways we could actually test those genomes in the lab.
This was no quick and easy task.
It was quite a long process of setting up this whole design framework where we needed to figure out a way, like, how do we actually generate a genome? Like, what should that look like? And how do we choose which genomes we ultimately will chemically synthesize and test in the lab? And what does it mean to design a genome was kind of the big question here. And it was a very creative challenge of coming up with all of the strategies and then little micro decisions on what that path should be and what the ultimate applications of it should be. And that was really the journey of this. And then we had developed this whole design framework of going from training the model further on bacteriophage genomes to actually developing certain methods to improve its generations on those genomes.
The last stage was to filter and pick genomes that met certain design criteria, and then setting up the protocols in the lab to test them at a large scale.
Once we actually started chemically printing these genomes and testing them in the lab, it was just a matter of getting one that actually worked. And we got 16 that worked. And so we were very happy with that and quite surprised as well.
This is brilliant. And I love the way everything kind of just collides. We're chemically printing genomes and using large language models to design them. And I'm going to actually need Samuel to convince me that this isn't ultimately going to result in just some catastrophic,
super scary stuff.
Super scary, AI creates viruses that eat everything, and it's paper clips all the way down.
So first of all, it was a very difficult process. It wasn't trivial. And so I think there's a lot of people who are concerned that, oh, this suddenly enables bioterrorism in worse ways or something. But as biologists, most of us can easily think of many other far easier ways that dangerous things could be done by someone with malicious intent. And using AI to generate some novel bacteriophage that's probably less fit than most natural bacteriophages and is not really the best way or most direct way to do that.
This study was much more of a research project in laying out methods for how AI models understand evolution and extremely complex DNA sequences at scale.
Where, the model, in order to achieve something like this, has to be able to reason about all of these long-range dependencies within these complex DNA sequences and at every scale, not just the single DNA base pair scale, but across whole genes or multiple genes and other elements that aren't genes in the genome. And I think this feat was a proof of concept in that we can now further use these AI models to really help study a lot more about genomes and also improve many biotechnologies that are genomic. And a lot of the most useful functions in biology emerge at the scale of an entire genome. In synthetic biology and biotechnology, we've been quite good at manipulating and engineering smaller elements like individual genes or maybe just a couple genes.
The team cannot yet do this reliably with genomes.
And it's because DNA is so unintuitive to us, and there's just an explosion of considerations we have to consider when we start increasing the amount that we want to engineer and design. And so here we have AI step in and help us with that process. And I think in that we are making a big step forward in allowing AI to really complement biological research and move us forward.
Samuel must have like an incredibly strong skull, because I would imagine his mind is blown every time he walks into a lab.
I know.
What does it mean to be a scientist today when things are moving so quickly? What's he learning about how science itself is actually changing?
It's crazy being a scientist today. Like I've been doing research for around 7 or 8 years now. And even just from a couple years ago or a few years ago, things have changed dramatically in biology, and largely due to so many advances in AI. We, especially those of us that do computational work and a lot of coding, AI has just completely transformed coding. We're able to interact with these AI agents that make it extremely easy to execute very complex code and just go from telling it a simple instruction to having a whole project laid out for you in just a matter of minutes, completely coded by the AI. And that allows us to do things at a speed way faster than ever before in analyzing data or developing new computational models or methods. But the slower side still is the wet lab work. And there still, I think, is a lot of development that needs to be made in making wet lab work faster, particularly in automation. And so for now, that's kind of a bottleneck for us, but there are definitely rapid changes happening in that regard.
This has got to be about a lot more than speed. It can't just be going fast.
Yeah, you're right. It's elevated ability to iterate on many ideas and have kind of this intellectual partner that you can constantly interact with and help you in your workflows. And in these more specific domains, like the biological language models that our lab has developed, this is allowing us to understand DNA in ways we never thought we could before.
By this time, these large language models have, of course, seen more genomes than any human has seen in history.
They can, train on millions and millions of genomes, whereas a single human could spend a whole life just studying one part of a single genome. And that's a huge advantage that we've gained from putting essentially all of observed evolution into one kind of universal model that has understand these principles around the fundamental units of life.
Some of this might make us think, we talked earlier about this sounds like science fiction, or it sounds like a Hollywood blockbuster movie. And Samuel actually has a nice comparison to the character in the Marvel movies, and he's thinking about Robert Downey Jr. as Iron Man.
Jarvis, you there?
At your service, sir.
Tony Stark is always talking to Jarvis, his AI assistant that helps him do all of these just insane ideas and create these new technologies that he probably would not otherwise be able to do on his own if he hadn't had this super intelligent partner that he's constantly interacting with. And maybe we're getting there with AI in the future. And right now we're seeing just the very beginning of it. And this is hugely beneficial to researchers and researchers who have, I think, very good taste in multiple domains and are very interdisciplinary and adaptable will benefit the most from the revolution that we're seeing.
So it's not a dystopian movie. It's a superhero movie.
In the sense that you can pick a superhero and you can compare, whatever you like to how quickly or how smart artificial intelligence is getting and making humans better. But this conversation actually made me think of Dash, who was the little kid from The Incredibles, which was so fun because Dash didn't know that he could run super fast, so fast that he runs on water. And at the point that he discovers this, he is running and he starts giggling. You were talking about—
It's a lovely little aside.
Yes, and
wonderful.
Is Samuel's mind blown, or does he giggle kind of like Dash on the water?
Sometimes it feels like that and you feel very empowered. Other times it's easier to feel a little bit more somber about it. And maybe sometimes you face hurdles that really set you back in your experiments or your research. But yeah, in general, it's definitely extremely empowering. And I think my PhD so far has been greatly accelerated by these tools. And I'm not even sure what it would look like if I had started my PhD even just a couple years earlier than I did.
Samuel said somber there, and it's got me thinking a little bit about the guardrails that his work on training choices, organism selection, the constraints that you have to put around experiments like this. What is his thinking around responsibility with these tools that could really reshape the fundamental basics of life as we know it?
Yeah, that question goes quite deep. I think there's a lot to carve out about that. I think at the everyday level, as scientists, we have a very big moral responsibility to pursue our research ethically and to always be thinking about what our intentions are. It's a dangerous thing to pursue any kind of research without thinking about what its ultimate implications will be for society or the environment. And so we should always keep in mind, okay, if I'm going to ask this question or pursue this idea, what will its benefits be? What will we learn from it? And it's kind of, I think, when you are so heads down, working hard and tunnel vision on something that you feel really passionate about, you can maybe get a little bit lost in what the bigger picture should be.
He talked about remembering to always take a moment to just step back and think, what is this all for?
We set out on this project really thinking about those impacts, and was a large reason why we, in the paper, did these demonstrations showing that the phages have a lot of evolutionary resilience and which could be beneficial for phage therapies.
We've talked today about superheroes and some striking potential comparisons, but Samuel says that it is really important to also recognize limitations in science and understand when humans should simply reach out for help.
And especially in these regards, like bioethics, biosecurity, biosafety, we should be consulting with experts who think about those things every single day and work side by side with them to improve the guardrails that we're implementing. And so I think not just doing it on your own, but really making sure that you're getting the help that's needed.
And we heard at the top that Samuel is studying at Stanford, of course, the beating heart of innovation in Silicon Valley. And I wonder if he feels the pressure of taking this mishmash of AI and biology and making it into a startup like so many other young people around the world must be doing all the time. I mean, is AI going to eat biology?
There's definitely a lot of pressure in that regard. Ever since moving to Stanford, there's so many people who want to pursue startups, and it's a huge part of the culture here. And it was one of the reasons why I chose to come to Stanford was I wanted to see the entrepreneurial spirit and maybe be able to become a part of it and pursue something like that in the future. And we've had a lot of interest in this project from particularly phage therapy companies. But I think for now, I'm not quite ready to pursue my own startup immediately on something like this. I'm still learning and figuring out what the most optimal thing to do is, and especially at a time like this when AI is just transforming so many things so quickly. I want to be very adaptable and flexible and very thoughtful about what I do next.
So there it is, folks. We've gone from just conceptually AI in biology in theory to seeing how actually they could fundamentally reshape the way that we study and adapt and drive things forward with the fundamental building blocks of life.
Absolutely. In that lab in California, they're taking computational models and lab experiments, bringing them together to tackle questions that used to take decades to explore.
And beyond the science, we've seen from Samuel how curiosity and creativity and really nice to hear a careful, ethical approach are just as important as the technology itself.
That's right. The future of biology is not just about new tools. It is about the people using them thoughtfully to push the boundaries of what is possible.
So that was fun. Guess what it's now time for? Every day is a school day.
It's a school day.
What have you learned this week, Dodie?
It relates to today's topic in the sense of us being imperfect and how we learn and what we can rely on when we learn it. And this comes from a book that I'm reading from 2021. It's the anthology of the best science and nature writing, edited by my favorite, Ed Yong. And we are chasing an interview with Story Collider. So if you're listening, Story Collider, we're coming for you.
We want you on the podcast.
We want you. And you know what else, listeners? We recommend listening to the Story Collider podcast because it's also great. But here is what Ed Yong says in the introduction to this anthology from, of course, you can imagine, 2021. A lot about the pandemic, but not all about the pandemic. Here he says, science is so much more than a library of publications or the opinions of doctorate holders and professors. Science writing should be equally expansive. Of the typical journalistic beats, science is perhaps the only one that draws us out of our human trappings. Culture, politics, business, sport, food, these are all about one species. Science covers the other billions and the entirety of the universe besides.
Oh, doesn't that just make you feel small?
This is so delightful. Oh my God.
That's kind of like the Carl Sagan pale blue dot piece, isn't it? It's
Exactly.
fantastic.
But isn't it glorious? Just a nice
What a
moment of
wonderful
humility
day.
for us.
Absolutely
What did you learn
brilliant.
this week?
Well, it's kind of like another piece of mind-blowing news, which does make you feel that we are a little bit small and not very important. So it turns out, do you ever see a film Gattaca?
Yes, I
About
remember Gattaca.
genetic engineering of children and so on. And of course, Gattaca, the movie, the name of the movie is using the 5 letters, the nuclear bases that make up DNA and RNA, A, G, C, T, and U. That's why it was called Gattaca, right? So, in other words, every fundamental unit needed to write life's genetic code is either A, G, C, T, or U, one of those nuclear bases. And guess what? Guess where they have been found? It's extraordinary.
Under the ice.
Nope.
Don't guess. Up in the air, in the trees, on another planet. On another planet.
Yes, that's it. It's it. Not on another planet. Until now, we knew we'd found some of them in meteorites, right? Not all of them. But we've never seen the full set together and never in such a clean, uncontaminated samples. They were discovered on samples returned from an asteroid. All 5 of those nuclear bases from a carbon-rich asteroid collected directly in space and brought back to Earth under super controlled conditions. And it just rules out the idea that the chemistry somehow leaked in from Earth. It's kind of an implication that perhaps maybe life came from space.
Yeah.
Maybe, but not really.
Wow.
But it just suggests that something, the raw ingredients for biology to assemble naturally in space long before planets even existed, that could have been there. Life isn't just a freak accident that happened once. Maybe it's somewhere else. So all very exciting.
Incredible.
Of course, everybody's speculating madly, but maybe life did start in deep space. Well, actually, life did start in deep space because we're also in deep space. So, I mean, it's a
Quite.
truism. Anyhow,
That's right. All right.
there you go. We learned some mad stuff.
Thank you for listening to this episode. Our producer, mixer, and get this, Conor, award-winning sound magician. Absolutely.
I love it.
Congratulations to Tom Henley. My name is Dodie Axelson.
And I am Conor McKechnie. Make sure you rate us on Spotify, whichever platform you use. Tell your friends and family and random strangers in the street about Discovery Matters. Thank you for listening.